Well, today we're going to take up the first of 3 modules on a subject that is fascinating to me and therefore is being inflicted upon you, and that is the story of the Black Death, the bubonic plague in medieval society back in the 14th century. So let's begin this discussion by reviewing or contemplating what would happen if you simply picked up the little insect we call the flea and maybe hold it up to the light. It's a curious looking thing. It's strange looking. It's almost comical in certain senses. If you do hold it up to the light, you'll see that you'll almost be able to see right through it. You'll see that it is extremely laterally compressed, so much so that it appears to be trying to operate in 2 rather than 3 dimensions. You'll see that it has no wings, and you'll see that it has these monstrous saltatorial hind legs by which it jumps up to 200 times its own body length. So think about that. Think about what that would mean for a human being if you could jump 200 times your own body length. You could probably, jump up to High Street in one bound. So it's a crazy looking animal, and because it is so odd looking, it just doesn't seem like the sort of thing that could be responsible for death on a massive scale, both geographically and temporally, and yet that's exactly what happened in the 14th century. The flea came to town, and with it, the story of the bubonic plague, something that mankind has been wrestling with ever since, and therefore is something I want to share with you in this course. It changed the very nature of society in the 2.5 years that the plague swept across Europe. It changed the relationship between man and God. It changed how we understand the world, and all of this is reflected in the kinds of ways that humans reflect upon society in art, in literature, in song, in dance, and so much more. And if you think about it, if you think what it would mean to you to go off to school or to work in the morning and then come home and find all of your relatives dead by dinner time, and this is happening not only to you but to many of the people around you, think about how that would change your conception of the world, the forces behind the world, the institutions that we use, all of us, to make sense of the world. Medicine couldn't save you. Religion was powerless. The kind of societal functions, politicians and whatnot, were blathering on about the problem, but completely unable to design a means of controlling the rampage of death that was coursing across medieval society. And as a result, this stands out as a profound moment in human history when all bets were off, when it really seemed as if the end of the world was approaching. And what does that do to the human mind, to the human heart, and to human interactions? That's the story I want to talk to you about in these 3 modules. So to begin with, we have to consider the protagonists. I've introduced the flea as the major point of interest, and that is true in the sense that it is the insect and therefore the thing that commands our attention in this course. But the complete understanding that I'm hoping you're going to acquire from this discussion is that it does involve a series of 4 species acting in concert. And without those interactions and those 4 species coming into proximity, we would not have had the thing that we call the Black Death, the bubonic plague in the 14th century. And so the overlap of these 4 species, as depicted in this lovely Venn diagram, which perhaps you recall from your high school days, is what I want you to be able to understand by the time we get through this discussion. So we're dealing with fleas, obviously. We're dealing with humans because, of course, we think we're the most important and therefore should be the focus of our attention. But also we're dealing with the lowly rat, the thing that runs wild in nature, and at least in medieval times was probably a fairly common kind of organism to find in the household of the average peasant. Most people back in medieval times lived in huts that had a dirt floor. Often these were in proximity, barns, and there was a fair amount of overlap between the animals, wild animals that existed in places like barns, and also in human habitations. So the rat will be important to our discussion. And lastly, the pathogen, which has a name. It is Yersinia pestis. That is the Latin name of the pathogen which causes the Black Death, the bubonic plague. So today we're going to be talking about the interaction of these 4 species and how together they produced the debacle that we know as the Black Death. We'll start with the flea. This is the biological pedigree of the flea. It is a proper animal belonging to the kingdom Animalia, and I don't really care that you know all of the different subclasses, but I will point out when we get down to the bottom of the chart where the genus and species are revealed that we're talking about, in this case, the rat flea, whose name is Xenopsilla chiopsis. We could be talking about a whole bunch of other fleas, because if you look at the family Pulicidae, which is the family to which the flea properly belongs, you would find that there are a number of species of fleas that might be of interest to us. There is an actual human flea, a flea that lives on people, which fortunately most of us don't experience in this day and age, but back in the days of the peasants living in huts, you can bet that some of them had human fleas. But it's not important to our story, which is really interesting, for reasons that we'll discover in a couple of slides, having to do with the biology of the bubonic plague and the very specific interactions that are involved in these 4 species that I mentioned on the previous slide, without which there would not have been a bubonic plague. So we're going to be focusing on Xenopsilla chiopsis, the rat flea, as a result of the aforementioned biology, which I will reveal to you over the course of this module. And you did see our lovely flea before I erased it. I just switched slides. Never mind. We'll just stay here. But if you go back to the previous slide and look at that picture of the flea, you can see the characteristics that I mentioned. The ability to see right through the darn thing. It has a pretty thin-looking cuticle. The lateral compression you can see, and you can see the impressive-looking jumping hind legs. Well, as you probably already can guess, the flea comes suited with a whole bunch of adaptations that it uses to earn its living as a blood feeder, ostensibly on rats, but as we'll find out, on humans as well. So it has specialized mouth parts that it uses for drawing blood from a living rat. It also has the ability to use carbon dioxide exhaled from the rat or from the human to locate that host. When we talked about the evolution of blood feeding, we went over this as a way of finding hosts, and indeed that's precisely how the flea locates its available host. I mentioned also that fleas are wingless, which is a really unusual condition for a proper insect, but because fleas have adapted so effectively to living on a mammalian host, which by definition will have a fur coat, or a coat of hair if you prefer, it has gone wingless over evolutionary time to avoid having its wings get caught up and injured by the hair. And so it just gave up the wings altogether, and that means, of course, that it can't fly. It can't get around by flying, as most insects can. And to compensate for that, it has developed an alternative method of ambulation, or getting around, and that would be those saltatorial or jumping hind legs that I mentioned previously. So fleas, if they need to travel, won't do it by flying. They'll do it by jumping with their impressive hind legs, and the other alternative way that fleas have of getting around is simply to hitch a ride on the organism on which they're feeding. So when a flea is living on a rat and blood feeding on a rat, it can move with the rat when the rat goes elsewhere, and that's a way for fleas to travel around and infest the whole darn neighborhood. What could be better? The flea life cycle also reveals some impressive adaptations towards living with vertebrates. For instance, the female flea will lay eggs directly in the bedding of its host. If it's living on rats, rats make little nests out of straw and debris and so forth, which they use to take care of their babies. And so the flea mother would be laying its eggs directly in that straw bedding or whatever the rat is using for the purpose of bedding. If the fleas are infesting humans, they would probably be taking up residence in mattresses and places like that, which would also be stuffed with straw and would be similar to the nest that the actual host or rat is providing. So it'll be hard to get away from the fleas because if you're a human, you have to take a rest every night, which we call sleep, and the fleas will probably be breeding directly in that bedding. Secondly, the eggs laid by the female will hatch in 1 to 10 days, depending on species and also on environmental conditions. Particularly when conditions are warmer, the fleas will hatch more quickly and develop more quickly. And so they can adjust their life cycle to the prevailing conditions. And if it's particularly cold, it takes longer, and they don't want to be emerging in very cold weather anyway. So this is yet another adaptation for them. When the larvae emerge from the egg, they eat detritus in the nest. So if we're talking rats, they're eating dander that falls off the rats, or scales from skin, or anything else that might be slightly organic in nature and could provide food to the larvae. If the larvae are in a human mattress, then again, your secretions, your sloughing off of dead skin cells and so forth can be used to feed those lovely flea larvae. And there will be usually 4 or 5 larval instars before the pupal instar develops. And here we have another important adaptation, and that is that the pupal instar can persist without feeding for up to 1 week to 6 months. And again, the goal from the insect's point of view is to wait until conditions are suitable for the continuation of the life cycle. But this is kind of impressive that the pupal form can stick around for up to 6 months without doing any feeding, meaning that it's going to be particularly difficult to get rid of these insects. And if you've ever had a flea infestation in your house, you know that that's true. You really have to be very vigilant to kill and control all of the life stages, otherwise you'll continue to have a flea problem in your house. Lastly, the adult female will take a blood meal from its host to produce the ultimate eggs, and that's one reason why fleas are blood feeders. And here's the last annoying but important factoid. The adults themselves, both male and female, can live for up to a year, and this is true even if they don't take a blood meal. And so, all told, the flea may take up to a year and a half to go through its life cycle, so it can be around for an awful long time, even if it's not taking a blood meal. So here's the basic mechanism by which the flea acts as a vector for the bubonic plague. We'll start in the upper left here, and we have, in this case, the flea taking a blood meal from a rat, the natural host. And if that rat is infected with the plague bacterium, the flea will take the bacterium into its mouth and then into its digestive system. And in the flea's digestive system, if we move to the second box here on the upper right, we find a very important biological thing taking place, which is absolutely critical to what ultimately happened in the bubonic plague. And that is that the plague bacteria will begin to multiply prolifically in the gut of the flea. And then we have another very important undertaking, which is, again, absolutely critical to what happened in the bubonic plague, and that is that the multiplication of those bacteria in the gut of the flea will result in the clogging of the digestive system of the flea. There are a couple of little tubes that ferry the blood from one part of the stomach to the next. We'll go over that in a minute with a diagram. And those thin little tubes can be clogged pretty easily when the bacteria multiplies. And part of that reason is that the outer coat of the plague bacterium is kind of sticky. And when it gets into this narrow area in the gut of the flea, the stickiness causes the plague bacterium to stick to the gut wall of the flea. And since we're talking about a narrow passageway, once the bacteria accumulate in large amounts, the gut itself is blocked. And this is enormously consequential because the blood that the flea is taking into its system is stopped in the part of the gut called the foregut. There are three components to the insect gut, you may recall from our discussions earlier. And the foregut is basically a holding area like the stomach is in the human. And in order to be useful to the flea, nutritionally useful to the flea, the blood has to flow from the foregut into the midgut, where the digestion begins, and lastly into the hindgut, where absorption takes place. But unfortunately, in the case of a flea infested by plague bacterium, the blood never gets to the midgut or the hindgut. It's stopped in the foregut because of the blockage that takes place to the thin little tube that I previously mentioned. And this means that the flea is not getting any nutrition, and although it's biting its host and taking in blood, it is technically starving because of the blockage of the gut. And what is the response to starvation? Well, the starving flea will continue to bite additional humans again and again, trying to get the nutrition that it is deprived of. And this is extremely important because a flea that is taking in blood contaminated by the plague bacterium will regurgitate a little bit of that infested blood back into its next host when it bites its next host. And that is how the contagion is spread from, we'll assume, a rat, which is what we started with, to a human. And this is likely because of a final aspect of plague biology, which is unique to this system, and that is the flea which bites the rat will cause plague in the rat. It will kill the rat. And that means that although we started with a live rat up here and the flea is drinking rat blood, by the time we get over here, the rat will have died and it has to go to another host because you can't pump blood out of a dead rat or a dead human for that matter. And so the rat flea now has the option or the necessity really to find a new host. And if it's killed all of the rats in the barn, it may very well move into the human habitation next door and find a human to bite next. And at that point, the plague bacterium gets transferred from the rat to the human. And that's how we get the bubonic plague. So it's important to realize that fleas are actually the first victim. And that is because of the blockage of the gut that I mentioned in the previous slide. The part of the gut that gets blocked in the flea is the little tubule called the proventriculus that marks the transition between the foregut and the hindgut. And of course, this leads to starvation of the flea because the blood is not getting to the place where it's digested and therefore can be used nutritionally. So the starved flea begins to bite more and more. Once all the rats are dead, humans become a perfectly acceptable host. There's nothing protecting them other than the fact that fleas and rats are most likely to cohabit. But once the rats are dead, the fleas are going to start looking elsewhere, and they're perfectly happy to take a human as a host if they can find one. And as you saw in the previous slide, that's how we get the plague bacterium from the flea to the human. So here we see at the bottom a lovely graphic of a grasshopper because it's a little bit easier to see than the flea. And what I want to call your attention to here is, this area, the crop, which is where the blood comes into initially and is held there for a while. But it's got to flow backwards into the midgut, which is here. And as you can see, there's this little tiny tube attaching the crop to the gizzard, and this is what gets blocked by the multiplication of the plague bacterium. So the blocked proventriculus is really what animates the sequelae, the subsequent events that take place that make the bubonic plague happen. It, of course, dramatically increases the transmission of plague because the flea is starving and it's going to keep looking for a source of food. Now, eventually the flea will die of starvation, but as we noted on one of the first slides, it can take up to a year for this to happen because those adult fleas are pretty hardy. The good news is the female who can't get a blood meal won't reproduce, but she can still continue to bite and spread the plague bacterium as her sole occupation at that point. We also know that there might be some temperature variables in the blockage of the proventriculus, the little part that I pointed out that gets plugged. At higher temperatures, there's less blockage, maybe no blockage, and so less transmission of the plague, so we'd expect to see some temperature variability in the plague, but that's a nuance that isn't necessarily borne out by the available evidence, so we'll leave that as a curiosity rather than as a fact. Now here you see the actual flea. Up here is the actual flea, and the authors of this paper have thoughtfully drawn in a graphic of the alimentary canal, and they have specifically pointed out the proventriculus, which is a little bit narrower in the grasshopper that we looked at, but it's in the same place anatomically, and this is where it is in the flea. And down here at the bottom, you see the flea, which is now loaded with the plague bacterium. This is the black stripe here is the plague bacterium. And you can see the proventriculus here, PV again, is swollen to a fairly significant size, and it's blocked, it's plugged up with the plague bacterium. Incidentally, I just want you to contemplate the reality that we know this, we have this picture because there are people in laboratories who are doing research on the bubonic plague in a laboratory with fleas. So, I'm surprised we don't hear, well, sometimes we do hear interesting stories about people doing biological research on the bubonic plague bacterium, one of which was on a guy, sort of one of these states' rights kind of guys who likes a lot of guns and is convinced the government's going to come and take his guns away. He was also a microbiologist living in Ohio, who ordered a vial of plague bacterium from the Center for Disease Control, and they nearly sent it to him until they investigated his background. So I guess that means you too could try to get plague bacterium, but maybe the criteria for releasing it are more strict these days than they were in the past. Let us hope. All right, let's move on to the discussion of the pathogen that causes the plague. a second protagonist, and we've named it already, as you learned. The bacterium Yersinia pestis is responsible for the production of the bubonic plague. It is kind of a wimpy bacteria. It's a so-called gram-negative bacterium. It is what we call an obligate parasite. It cannot live outside the host. So if this bacterium were released into the broad environment, it would die fairly quickly. It would not contaminate surfaces, which if you touched, you'd get the plague. So that's not a mode of transmission at all. But we also know that Yersinia pestis is a bacterium in a suite of related bacteria, all of which belong to Yersinia species. And I want to mention this, not because we expect you to be an expert on the genus Yersinia, but because the genome, the DNA of Yersinia species seems to be pretty malleable, pretty susceptible to change. And we know this because the virulence level of the bacteria in this group is highly variable. We also know that there are some novel genes in the particular species of interest, in Yersinia pestis, that are in there, in the Yersinia pestis, which help Yersinia pestis overcome things like antibiotics. In other words, it has genes for antibiotic resistance, and there's a fair amount of coevolution going on between the bacterium and the available hosts. So let's talk about that a little bit more. We know that Yersinia pestis, the one that causes bubonic plague, evolved from a related species, Yersinia pseudotuberculosis. So those two split off about 20,000 years ago, which is pretty recent. We also know that there are 3 identifiable subtypes of Yersinia pestis. They are Antiqua, Medievalis, and Orientalis, each of which was associated with a major plague outbreak among humans. So Antiqua is the oldest one, taking place in the, was it the 5th century, thereabouts. Medievalis is the 14th century plague, and Orientalis is a more recent plague, but taking place in the Orient, hence the name. So we have a fair amount of ability now to analyze directly the DNA of all species of Yersinia, and we've been able to establish how these species evolved one from another by looking at DNA analysis. We also can do DNA analysis on victims of the plague in modern times. There have been a bunch of arguments for quite a long time, more than a century, about whether any of the plagues referenced in this slide were in fact due to Yersinia pestis. And the arguments ranged from various points of view, but the availability now of DNA analysis has really enabled us to go back and find out whether the people who died during these different pandemics were in fact killed by the bubonic plague. And one of the ways they can do this is they can take teeth from the victims. The teeth are a great way to look for plague because when you're alive, the teeth are connected to the bloodstream, so the plague bacterium can get into the internal parts of the teeth. But once you die, the teeth are sealed off from the environment. So whatever's inside the tooth, even if it's 150 years later, is a pretty good representation of what was going on in that person at the time the person died. And so people have gone through medieval graveyards, for instance, and found people who died during the time of the plague and analyzed the dentin inside the teeth and discovered Yersinia pestis, indicating that that person did indeed die of the plague. So we have a lot of DNA analysis at our disposal in modern times. And another thing we've done with that DNA capacity is to look at the genomes of the related species of Yersinia to determine their evolutionary relationships one to another. So using that kind of information, we know that, as I already mentioned, Yersinia pestis came to us from Yersinia pseudotuberculosis. We also know that there's a third species in this group, that is Yersinia enterocalyptica. And the interesting thing here is that if we compare the three species, we will find that both Yersinia pseudotuberculosis and Yersinia enterocalyptica, both of those are capable of causing human illness, but they cause basically a stomach ache. They're gut poisons. They make you feel like you've got the flu because they're impacting the intestines. In contrast, Yersinia pestis causes the plague. So if you look at these two different groups, the gut poisons, which is the ancient modality, and you compare it to Yersinia pestis, which is a comparatively new development in this group, we find some really interesting things. First of all, the ones that cause the stomach ache, the first two species, do this through the so-called fecal-oral route of contamination. So to get one of these bacteria, what happens is the bacteria are shed in the feces of an infected human. If you touch those species, and there are various ways of doing that, believe it or not, which is why we tell you to wash your hands after you go to the bathroom, or before you prepare food. If you come into contact with those feces and touch them with your hands and then touch your mouth with your contaminated hand, the bacteria can get into your gut and then rummage around in your intestines and cause the stomach ache that I referred to. And now the bacteria will be inside your gut, and you will shed those bacteria when you defecate, and so you can spread the joy, as we say. But the important thing to note is that, first of all, there is that fecal-oral route of contamination, and secondly, the stomach ache that these two species of Yersinia cause is rarely deadly. It's unpleasant, but it doesn't kill you. In contrast, we have Yersinia pestis, which has an entirely different mode of transmission, which involves having a blood-feeding insect bite an infected host and then bite an uncontaminated host to spread the bacteria. And we have a level of virulence with Yersinia pestis, which is highly lethal. And what is most disconcerting is that this change from the fecal-oral route to a blood-borne route, and also from low virulence to high virulence, took place very rapidly in an evolutionary time frame. And that is concerning because who knows what additional changes might take place somewhere down the line that might even further accentuate the danger caused by Yersinia pestis. It looks as if the genome of Yersinia pestis is sort of set up to be mutated and for those mutations to be passed along to offspring. I mentioned previously also that Yersinia pestis has the capacity, not all versions of Yersinia pestis, but some are already resistant to antibiotics. Antibiotics are the major way to control Yersinia pestis. Of course, antibiotics were not available in medieval times, and so the disease basically just had free rein to course across humanity. Now we would treat a plague victim with antibiotics. But bacteria in general, and Yersinia pestis in particular, are pretty adept at becoming resistant to antibiotics and also spreading the genes for that resistance. And the major mechanism by which they do that is through the plasmid system. In fact, most bacteria have small bodies inside the bacterial cell called plasmids. And in the plasmids, they store important pieces of DNA that code for survival traits. And among those survival traits are genes for pesticide or antibiotic resistance. So it's a really important thing that Yersinia pestis has these plasmids, and that we know that within these plasmids there are already genes that can reduce the effectiveness of things like antibiotics. We also know that there are, because we've now been able to analyze the genome of Yersinia pestis, as well as the DNA that's contained in plasmids. And we know that there are particular genes within the plasmids that, for instance, increase the pathogenicity of Yersinia pestis to humans. We also know that there are specific toxins. We call these murine toxins. Murine is the Latin descriptor for rat. So there are particular toxins in Yersinia pestis that are highly lethal for rodents and are relevant to us in this discussion because as the rodents die, we've already mentioned that the fleas go looking for humans as a means to get that blood meal that they're lacking. But you have to be impressed with the fact that although the bacteria, Yersinia pestis, is kind of a wimpy bacterium, it nonetheless comes armed with genes that help it avoid being intoxicated by antibiotics and also increase the lethality of the subsequent disease to the host, both human and rat. And so, as I indicated on the slide, when the plague comes, all of the organisms in the dance, all of them suffer, except for Yersinia pestis. Here's a graphic of the bacterial cell. Up here you see the bacterial cell with a nice thick bacterial cell wall. And inside you see the one circular chromosome of the bacterial cell. This is the main DNA that it has. But also over here you see these little tiny plasmids. And within the plasmids you will also find DNA that can code for the special genes of interest, particularly antibiotic resistance and virulence genes. The thing I want to mention here is that it's possible for two bacterial cells to swap and exchange plasmids. And to do this, you would have two bacterial cells lining up next to each other. They would form what we call a conjugation bridge between the two bacterial cells, and they would pass plasmids between the cells through the conjugation bridges. So there is an inherent mechanisms by which bacterial cells freely exchange plasmids and consequently the genes that are contained in those plasmids, which may have important impacts on disease transmission and virulence. Yet another thing of interest in the biology of the bubonic plague. We haven't talked much about the rat flea, other than to mention that it's a victim and that it's important in the transmission of the bubonic plague, but let's reiterate that. The rat flea is interested in finding rats to get a blood meal. But as we've mentioned, because of the peculiarities of the way Yersinia pestis behaves in a flea, particularly the accumulation of the multiplying bacterium in the gut in a way that clogs the gut, what this means is, first of all, the rat gets sick from the disease. Subsequently, because of the high virulence and the murine toxins that I mentioned a few seconds ago, the rat dies. The flea cannot withdraw blood from a dead rat. So the flea will move on to another mammal, and because, particularly in medieval times, humans and rats were basically cohabiting, humans became the next best and indeed likely choice to be the food source for the flea, now starving, even though it's repeatedly biting and withdrawing blood from victims. And in the process, as it turns out, spreading the bubonic plague. So, it gets to humans. And now it's time to talk about another wrinkle in the biology of the bubonic plague that is enormously consequential and also a little confusing. And if it's confusing to you now in the present day, consider how baffling it must have been to physicians in the time of the plague. One of the reasons for this is that humans can potentially acquire the disease that we're kind of collectively calling the bubonic plague through 3 different routes of transmission. we've already talked about the bite of the infected flea, and that is indeed probably the major mode of transmission and the one that is responsible for the movement of the plague from rats infested by fleas to the human population. But once a human has the bubonic plague, sometimes, not all the time, but sometimes, the plague bacterium moves to the lung of the humans and begins to replicate in the lung tissue. And that means that every time the human exhales, it's possible for plague bacterium to contaminate the air that the human expires, and also the saliva of the human, because the plague bacterium, as it's on its way out of the body through the breath, can end up in the saliva of the person in his or her mouth. So this form of the plague that inhabits the lungs is called pneumonic, and it is one of the few ways in which you can get transmission of plague directly from human to human without the intervention of a flea. Now, it turns out that this particular form of plague is a little bit more severe than the bubonic plague, which is spread only by fleas, because the pneumonic plague attacks the lungs and the respiratory system. It is inherently more fatal, and even more importantly, and I cannot emphasize this enough, because you can get direct person-to-person spread of the plague through contaminated sputum and contaminated saliva, this is a major way in which plague spread in medieval times. Again, without the intervention of fleas. Now let's go over in a little bit more detail the particularities of the three types of plague. I've already mentioned two. We'll talk about the third in just a moment. The bubonic plague, the thing that we refer to as the bubonic plague, is so called because once the plague bacterium gets into the human blood system, one of the first stops the blood takes in general circulation is the lymph nodes. The lymph nodes, the lymph system, lymphatic system, and the blood supply are intimately connected. And it turns out that in the case of humans, the plague bacterium is very likely to multiply in those lymph tissues and lymph nodes. And the reaction of the lymph node to being infested by bubonic plague bacteria that are now replicating in the lymph nodes is to swell up. The swelling is a combination of things, extra tissue, but a lot of fluid buildup inside the lymph nodes. And this results in the eruption of what is called a bubo, or buboes, plural. So the lymph nodes become inflamed, they swell, and that is, generally speaking, the first sign one sees of bubonic plague in humans. There can be a lot of tissue damage underneath. There can be some blood draining into the buboes, and this tends to make them black in color, really ugly and scary looking. And so the eruption of those nice black buboes, which are, first of all, painful, but secondly, a sign of a dread disease, are usually the first indication that a human is suffering from plague. Because this kind of plague transmission is associated with inflammation of the lymph nodes, which we call buboes, it is therefore referred to as the bubonic plague. The second type, the pneumonic plague, can happen in a couple of different ways. It can happen directly from a person who has pneumonic plague and breathes into the atmosphere bacteria that another person who's not infected takes in when he breathes. And having taken plague bacterium into his lungs, now the plague bacteria may begin to multiply there and result in primary pneumonic plague. Now, the reason this is different from the bubonic plague even though it's caused by the same bacteria, is that you typically don't see the eruption of buboes because it doesn't go first to the lymph system. Its first stop is the human lung if you breathe the bacterium in. So think about medieval physicians are trying to deduce all this. And you got a really sick person with pneumonic plague, but you don't see any buboes. So you're thinking, well, it's got to be something else because obviously it's different. And yet it's not, but they didn't know that at the time. And the final form, the third form of plague, is the so-called septicemic plague. And this is a fairly rare condition, but what happens is the bacteria enter the human circulatory system through the bite of a flea, and instead of progressing immediately to the lymph nodes and multiplying there, instead the bacteria begin to multiply in the blood. So it's probably a genetic difference among humans that makes this happen in a small number of people. And people in whom this happens get the septicemic form of the plague, which is the most deadly of the three forms. So you've got three different forms of plague with three different symptomologies and three levels of virulence, making this a real mishmash of confusion for physicians who are trying to figure this out. And here I've given you a table that attempts to summarize all of this, which I'll go over. We have the different forms of the plague here. You'll see that there are 5 potential different forms. This column summarizes whether or not you see buboes in the course of the disease. We also look at whether or not you can get direct human-to-human transmission. If you can't, then it means that you have to have the involvement of a flea in order to do it. I've given you the mortality rate of each form during the medieval plague, with the caveat that if you check different authors, I looked at Butler, 1995. If you check different authors, you'll find different mortality rates. So I probably won't ask you questions about the mortality rates of the plague because it's all over the map. And also I gave an assessment of the number of percentage of total cases for each different type of plague. But again, these are estimates, so I don't put a lot of faith in the data. So if we look at regular old bubonic plague communicated by the bite of an infected flea, yes, you do see the eruption of buboes. That's usually the first sign of a problem. And you cannot get direct human-to-human transmission of the bubonic plague. If it's just in the lymph system, you will not get direct human-to-human transmission because that has to come from the lungs. In medieval times, the mortality rate of the bubonic plague was pretty low, 60 to 80. You'll also see other authors who claim it was 30 to 40. I am not in a position to dispute it one way or the other, but this is a range quoted by one author. If we then get into the different kinds of plagues, we talked about primary pneumonic as taking place when you have a person already infected with pneumonic plague and he or she exhales, spews plague bacterium into the environment, and another person takes those bacteria in to his respiratory system. That's when you get primary pneumonic plague. You don't see the eruption of buboes, making it confusing. You can get direct human-to-human transmission without a flea. The mortality rate is distressingly high, up to 95% during the time of the Black Death. Oops, I just told you the wrong, oh, no, that's right. Okay. Secondary pneumonic plague occurs when you have regular communication of the bacteria from a flea bite, and the bacterium invades the lymph system as per usual, but the replication of the bacterium takes place not in the lymph nodes, but instead in the lungs. So it starts out as what should be a bubonic plague case, but it ends up going to the lungs secondarily, where it becomes a pneumonic form of the disease. Because it's pneumonic and because the sick person is now going to spew this into the environment, you can get direct human-to-human transmission, and again, a very high mortality rate. Then let's move into the septicemic category. The difference between primary septicemic and secondary septicemic is this. In the case of primary septicemic, what happens is, and this must be due to the genetics of the person getting the bite of the infected flea. In the case of primary septicemic, you don't see the eruption of buboes. The bacteria gets into the bloodstream. It should be able to travel to the lymph nodes and to the lungs, but instead of replicating in one of those sites, it replicates preferentially in the blood. Therefore, you don't get buboes, nor can you get direct transmission to other people because it has to go to the lungs and, replicate there, which it isn't doing. However, it is replicating in the blood, resulting in a very sick person with, in medieval times, 100% mortality rate. Secondary pneumonic comes about as a result of somebody who gets genuine bubonic plague, so you do get the eruption of buboes, and just to add insult to injury, the bacterium starts replicating in the blood, and you also get a septicemic form in the same person, again, a very high mortality rate. So I give you all this information not to confuse you, although I probably did, but more to point out how difficult it must have been for medieval physicians to look at all these different modes of transmission, all these different symptomologies, all of these different mortality rates, and try to figure out, oh, we must have 4 diseases going on here or something. And of course, in medieval minds, there would have been interest in finding different cures for the disease based on the different symptomologies and so forth, and it just compounded an already difficult situation where ignorance reigned and superstition was the palliative most often used to fight not only the plague but most other maladies. And on that point, where did people in medieval times think the plague came from? Because they knew nothing about flea bites and transmission of bloodborne pathogens. In fact, they didn't even have the germ theory of disease, so they didn't even know that germs, these invisible things that fly around in the air, can make you sick. That was completely unknown. What they thought in medieval times, and the reigning idea that had been out there for a couple of millennia, was that disease was the result of exposure to a miasma. What is a miasma? Well, it's a big cloud of toxic or poisonous air. Most miasma were thought to settle or occur in low-lying areas where moisture can accumulate. Miasmas were associated with a bad smell, a stench, as would be typical in low-lying swampy areas. And the miasma idea was particularly attractive to medieval people and earlier people because, as you know, air can move around. And so this is a mechanism for explaining why and how plague and other diseases could move from group to group of people. So it was the miasmatic theory that reigned supreme during the time of the Black Plague, and indeed for quite a while thereafter. And while it was useful because humans are very keen on establishing causation for bad things that they don't understand, it wasn't particularly helpful in terms of deducing causes of the disease and treatments for the disease. So the evidence that the miasmatic theory of the plague was what people were looking at is pretty well known because of discussions taking place during the time of the plague where people were thinking about, there was that weird arrangements of the stars or planets last week, and that's when the plague hit, and so people went to a lot of effort to try to argue that astral arrangement of stars is what caused the miasma, and that's what resulted in the plague. So you can find those kind of arguments and justifications in medieval literature. So once the plague took root, it had immediate impacts on society. Here you see, just as an entree to this discussion, which we'll take up in earnest in the next module, an early woodcut that found its way into the Toggenburg Bible. And what you see is a couple of parishioners in the foregrounds who are literally riddled with buboes, looking very, very painful. And the feckless priest in the background looks like he's spilling magic fairy dust in the atmosphere to try to dispel the plague. And I'm sure these people simply just died. So let's take an overview, sort of a panoramic view of the plague as it coursed through Europe. The dates of the plague are, it probably reached the Mediterranean area in Crimea by around 1346. It's thought to have arrived from Asia, probably as a result of the Mongols, who were busy establishing trade routes, both overland and then later by sea. The Mongols are probably the source of the plague because they were operating in Asia, and there are graveyards in Asia immediately preceding this time that show massive pandemic diseases. We don't know what the symptomology was particularly, but it's likely that those pandemics reflected in graveyards and tombstones and so forth were the source of the original plague, which was then taken up by Mongols, and because of their desire to expand trade routes and also their basic nature of conquest and desire to acquire more land and so forth, probably were responsible for spreading the plague from Asia via overland routes throughout the Asian countryside, and probably there were additional characteristics of the Mongols that made it more likely that they would do so. For instance, they were fairly truculent people. They were interested in conquest. They had a number of battles pretty much all the time, and their habit, once they had dispatched a population, was to then take all of the valuable goods. And if the valuable goods included pelts, which they probably would, and those pelts had on them fleas, now with plague bacteria, then they would have a mechanism for accentuating the rate at which the plague progressed across Asia and then ultimately into the Mediterranean area. So once the plague reached the Mediterranean, there was a second mode by which it was transported, and that was via ships. And I can document that for you in the book, where there's a pretty good discussion of how the Mongols reached Kaffa in the Mediterranean and tried to storm a fortress to take it over, but were prevented from doing so because they themselves had the plague. But rather than let the people inside, who happened to be Christian, stay there and continue to fight them, what they did was catapult plague-riddled bodies over the ramparts inside the fort, so the people inside get plague. And then that became a way by which the people inside who left the fort, realizing now that it was a source of disease, would then help to transmit plague throughout the countryside. Plague was also reaching port cities in the Mediterranean via ships. And this is documented in a variety of chronicles. Ships were described as arriving unmanned, that everybody on board had died at their oars, as was the phrase used. And this meant that once the ships pulled into port, now with a entirely dead crew, the people in the port cities would want the goods on board the ship, and so they would enter the ship and take the stuff that was on board. And again, they would expose themselves to fleas that had come over on rats with the boat. And, of course, they would carry the stuff up onto the shore where fleas would then spread it to the indigenous population in the port city. In addition, rats come and go from ships, so rats still living but infested with fleas carrying the plague bacterium would also spread the joy in those port cities. And for that reason, the plague basically followed trade routes across the Mediterranean and into European cities. Once the port cities became infested with plague, people began to move inland, trying to outrun the plague, and also to the north, which again simply spread the plague even more, causing great devastation in social, religious, and economic terms as the plague moved across the landscape. Now, one thing that people generally don't know, but was certainly true, is that the bubonic plague lasted maybe 2.5, 3 years, depending on how you want to count it, and then it went away. But it recurred in Europe a couple more times over several centuries. And one of the interesting things was that as it recurred, the virulence, the death rate, was lower each time. And by the 19th century, Europe has been plague-free. So it's just a strange disease in so many ways. As I mentioned, the way you know you have, or you probably knew that you had the bubonic plague back in medieval times, was that you got those lovely buboes. It's a very painful disease. The swelling is usually in the neck, the armpits, or the groin, because they are swollen, they hurt. Every time you move, it hurts. And so the pain caused by the buboes alone would have been a formidable thing to deal with. But there are a number of other symptoms that accompany the disease, depending on which form you got, but they include fever, headache, painful aching of joints, vomiting. Sometimes it gets into the nervous system and causes hallucinations and so forth. It's kind of an all-purpose invading disease that attacks in multiple different ways. As I mentioned, the bubonic form was the form most commonly seen during the period we call the Black Death, with a mortality rate that, if you sort of look at the extremes of the ranges reported for mortality, would be between 30 to 80%. And a typical course for the bubonic form of the disease would be that you'd get sick. That would last for about 4 days, maybe 7 days if you were, I guess I could say you were unlucky at that point, because it's a pretty nasty disease. And then you die. So you're sick for several days and then you die. The other forms tend to kill you more quickly, and we've already reported on the virulence rates or the mortality rates for those forms. So the pneumonic form is the second most common form, and the symptoms are somewhat different. They include fever, which is the case for bubonic, but also that cough and the blood-tinged mucus, which of course is enormously helpful in spreading the disease without the intervention of a flea. We've already indicated that the mortality rate was in the neighborhood of 90-95%. And lastly, the septicemic form, which fortunately is the least common, but has some different symptoms, including a very high fever, purple patches on the skin, which may be due to a couple of different things. They're not quite sure why, but you can imagine having purple skin would make you look pretty horrific. And of course, that was a very scary form to see, if only because in medieval times the mortality rate was pretty much 100%. Now, as the plague marched through history in Europe, again, the numbers vary, but somewhere between 45%, 50% of the European population was killed in a 4-year period. There was variation according to geography. It may relate to the temperature, as we discussed earlier. But if so, the temperature worked in a strange way. The colder regions looked as if they had a lower mortality rate. So I don't put a lot of stock in this information. Where did the plague come from? I addressed that a couple of minutes ago, but probably Central Asia was the ultimate source. We know this because of graveyards with massive numbers of graves that come into being very quickly in a short period of time. And a Russian archaeologist, Chwolson, has done a fairly extensive search of those graves and has determined that the graves are reporting death due to plague. Again, I mentioned that plague moved overland due to the Mongols and their activity, arriving at the port city in Crimea at Kaffa, and the events that took place with the ramparts being breached by plague-ridden corpses helped to spread it inland there. From Kaffa, it was a short distance for ships to move from that city to places like Sicily and Venice, and thence to the Italian mainland, where, well, you know what the result was, moving north and inland after that. So here's a nice graphic of the arrival of plague. Here we have Sicily, some of the cities down here, and then it was taken from these places to port cities in the Mediterranean region, and then elsewhere via overland routes, and also continued distribution by ships going to distant cities along the coast. So you can see that the plague, if it arrived in the Mediterranean, in late 1346, it was showing up in these cities in 1347, moving north and inland until 1350, when it subsequently died out. There is an interesting debate, which we'll get into more in the book. I won't regale you with it too much, because there's an entire book written denying that the medieval plague was due to the plague by a guy who thinks that the wrong rat was present in peasant houses to provide a vector for the plague. That was his argument. There are other arguments that also look at other pathogens, but as I've said, most of those have been disputed because of the availability of DNA evidence in the form of plague in people's teeth. But there was a standing argument also about, well, if there's plague in Europe, there has to be what they called a sylvatic reservoir for plague in the wild, meaning there had to be a wild rodent somewhere in Europe that was responsible for harboring the plague bacterium and thus fueling the subsequent outbreak of plagues. And because they couldn't find one, the argument then again came back that, well, it couldn't be the plague, it couldn't be the bubonic plague, because there's no rodent in the wild that would keep the bacterium going to support subsequent plagues that did take place in Europe. And so there's a paper by Schmidt and his colleagues in 2014 that took this issue on and asked the basic question, well, is there a wild European rodent that harbored the plague bacterium and made it possible for the subsequent European plagues that took place after the original bubonic plague? Or was this instead a case of multiple reintroductions from Central Asia? And so to figure that out, what they did, what Schmidt did, was to scan the European countryside. Now, I'd like to have a project like that. Send me to Europe for months at a time to walk around in the countryside and look for rodents and figure out whether they could harbor plague. So he did that, and he couldn't find anything obvious. And so then he went to Central Asia, and it turns out that there's a wild giant gerbil. I'm sorry, I'm laughing. Gerbils are funny little animals, and that they should be the source of a very deadly disease is very strange to me, but it turns out the wild giant gerbil is probably the source of plague in Asia, and there is evidence that plague moved from the giant gerbil to Europe several times, and is reported in this paper. So that's probably what happened during the time of the Black Death. There were multiple reintroductions of the bubonic plague from the Central Asian routes where it is endemic to the area. It is not the case that there are rodents in Europe that are harboring plague. It's probably a case of multiple reintroductions from the Asian sources. So that's what we know about that. And here's your study guide. There's a lot of stuff on there, and we'll be taking up elements of it in the 2 subsequent modules that deal with the bubonic plague. Cheerio!